Have you ever walked outside and asked yourself the question, where did this all come from? I know I have. Well, if you're like me, then you'll want to stick around to finally receive the answers that you've been searching for. For decades, people have been trying to find the truth behind the question, where do we come from? Scientists known as paleontologists quickly began to try to uncover the truth by gathering and analyzing fossils. We live on a very large and very old Earth. Because of the many continuing evolutionary changes on Earth, when searching for fossils and other evidence from the past, paleontologists were able to find a very large variety of fossils, many different species, and also many from many different places and decades of life. In order to fully describe the many differences amongst the many different species living on Earth, scientists had to come up with a geologic timescale to divide Earth's history into a series of eras. With the geologic timescale, scientists were able to infer that modern species have evolved from extinct ancestors. In the 60s and 70s, scientists began to analyze the DNA and proteins of different fossils, and were later able to explain how different species are related to one another. So that deep connection that you may feel with your at-home pet may be just a little deeper than you thought. Interesting. Very interesting, I'd say. In this segment, we will be speaking with specialists to examine the different approaches to studying the evolutionary changes within both living and extinct species. Thank you for coming to our show. This is our first guest, Jenny Lynn.
Thank you for having me. I'm happy to be here.
So, Jenny Lynn, tell us a little bit about your field and why you're here today.
I was asked here today to talk about fossils and how paleontologists and scientists have found evidence from fossils to support our origins and evolution, among other things.
Oh, okay. So what exactly is a fossil?
A fossil, to start, is any evidence of an organism from more than 10,000 years ago. There's a wide variety of shapes, sizes, and vary from dinosaurs to humans to plants. And some of you might remember from the movie Jurassic Park, fossils can also be found in hardened sap, like the mosquito that found dinosaur DNA still in its stomach, allegedly.
Of course, what a classic. I remember that.
Some places were once completely covered by water, and that was discovered by finding fossils of creatures of the sea, which is now on dry land. So at some point, the land had been completely submerged.
So interesting. So I'm curious, how are fossils made?
Well, there are many ways the fossils can be formed. For example, compression, which is when a leaf sinks, then a fine sediment like sand covers it, and the imprint or fossil of the leaf is found inside, compressed for thousands of years. Then there's petrification, which is when a human or animal is decayed and buried. Then the water containing dissolved minerals seeps through and hardens the earth around the bone. And when uncovered, the bone is still intact. Third, there is impression, which is when an animal dies, for example, in the mud, and then decays like petrification, and the mud hardens to rock, and the fossil is uncovered under the rock hard mud. And then last, there's cast, which is made when an animal dies but sinks into a soft area like the bottom of the ocean, and it turns hard like rock. Then a cast is formed around the decayed animal.
Very informative. So can you tell us how old fossils are?
Sure. Like I said earlier, to be considered a fossil, the organism has to be at least 10,000 years old. So to predict the age of a fossil, it is tested in two different ways that are pretty complex, but I can sum it up for you, Christine. First, there's absolute dating, which refers to the dating of fossils or artifacts from a particular site. And there are different ways of coming up with this date, but it has to be a specific date. It cannot be a guesstimate. And second, radiometric dating, which is the method of dating geological or archaeological specimens by determining the relative proportions of particular radioactive isotopes present in a sample.
Very complex. Do you think that the study of fossils relates to evolution and tracing our own fossils from where we originated?
Absolutely, Christine. The study of our Earth's history is so relevant for our knowledge and research to help better understand where we come from and how we evolved. By studying our past, we have discovered evidence of our origins dating back to originating from primates. Charles Darwin's theory on natural selection explains how we humans had to adapt and evolve in order to survive. With today's technology, we have just scratched the surface of what will be uncovered by today's paleontologists and scientists.
Wow, this is also very exciting. Well, thank you so much for sharing with us.
It was a pleasure. Thanks.
Now we will transition to our next guest, Gavin, with Muhammad in the studio. How are you guys?
I am here with Gavin, who wants to share with us something exciting.
Thanks for having me on the show.
What did you want to talk about today?
Since the topic today is evolution, I'd like to talk about biogeography and the role it plays in evolution. Biogeography is the study of the role geography plays in biology. I'll explain. A good place to start is with the theory of plate tectonics. Plate tectonics is very relevant to the fossil record, so it's important to know about when considering biogeography. Layers of the Earth's crust, known as tectonic plates, have shifted over hundreds of millions of years. This process has not stopped. According to this theory, all of the continents were originally part of one supercontinent called Pangaea. The continents, as we know them today, sometimes share a tectonic plate. The theory of plate tectonics helps to explain the sometimes confusing distribution of fossils across the continents.
Could you give us an example to help us understand?
Of course. Here's an example. The Indo-Australian plate is shared by the Indian subcontinent and Australia. Fossils of ammonites, which are a type of large mollusk, were discovered high in the Nepalese Himalayas. Nobody could explain the presence of marine fossils thousands of feet above sea level until the theory of plate tectonics was postulated. According to plate tectonic theory, the Himalayan mountain range was created as a result of the Indo-Australian tectonic plate colliding with the Eurasian plate. What was once the sea floor was shifted thousands of feet up by the force of the collision. This is how the Himalayan mountain range was formed. So, the fossils found up there are relics of this ancient cataclysm.
That's fascinating.
Indeed it is. Another helpful example is that of the marsupials. Researchers discovered that marsupials were the most proliferous land mammals on Earth until about 110 million years ago. Around 110 million years ago, placental mammals, which develop much more than marsupials do before they're born, began to displace marsupials.
How did the marsupials go from being so widespread to being so geographically limited?
That's a good question. Because marsupials are born in an utterly defenseless state, they made easy prey for other animals, such as the placental mammals, and began to disappear from many regions. The fossil record indicates that marsupials lasted much longer in South America. Of course, they are still widespread in Australia, which I'll explain shortly. Researchers propose that prior to about 65 million years ago, both North and South America were completely separated by water. It followed that marsupials remained the predominant mammal in South America for so long because placental mammals had not evolved on that continent, leaving the marsupials in peace. From 65 million years ago on, the Isthmus of Panama began to form from the eroded sediments of the two continents. Once enough land had formed to connect the two land masses, the placental mammals in North America could cross into South America and prey on the marsupials. However, Australia became separated from Antarctica around 60 or 70 million years ago, becoming isolated from the other land masses, so the marsupials were able to survive.
That's amazing. Are there another example you can give us?
There are other biogeographical phenomena worth noting. Island biogeography is a unique factor in evolution that affects species on a similar but smaller scale as continental biogeography. A phenomenon called adaptive radiation occurs when a species moves from the mainland to an island. Because of the unique conditions of the island, the species evolves differently than the same species on the mainland. One of the most famous examples is the diversity of species on the Galapagos Islands. Another concept in biogeography is Wallace's line. Alfred Russel Wallace, a British naturalist often forgotten in the shadow of his more famous contemporary, Charles Darwin, noticed interesting differences of species distribution in the Malay archipelago. He noticed that marsupials tended to predominate east of a line through the islands, and that placental mammals appeared more west of the line. It was later discovered that a deep water trench existed where the line was imagined to be, which kept the islands on either side separate for millions of years. Because this natural barrier prevented most species from migrating to islands on the other side, the species developed separately. These examples illustrate the enormous role that geography plays in biology. The shifting of land masses over hundreds of millions of years has profoundly affected the survival and distribution, or the extinction and disappearance, of many species.
You also wanted to say a few words about anatomy and evolution, didn't you?
Yes, I did. Another field that helped to explain the evolution of species is anatomy. As we know from Professor Hermoso's lectures, all life, whether prokaryotic or eukaryotic, is based on the cell. When we consider the anatomy of the whole body of various organisms, we can find clues to the common ancestry of all life. In this case, let's look at multicellular animal life. An anatomical structure in one species that's similar to the anatomical structure of another species and reflects a common ancestry is called a homologous structure. An example of such a structure is the vertebrate skeleton. The study of homologous relationships between species can be useful for many purposes, such as in genetics and the identification of fossils. Although homologous anatomical structures share a common origin, they do not always have the same function. Our middle ear bones, for example, originally were supportive structures for the jaws of primitive fishes. Although the middle ear bone does not serve that purpose today, in humans, it does reveal a shared developmental ancestry.
Aren't those called vestigial structures?
Yes, that's right. I'll explain. A vestigial structure is an anatomical feature that has no apparent function in one species, but is homologous to a functional organ in another. For example, some whales and snakes retain bones such as a pelvis and femurs that no longer function for their original purpose. These vestigial structures are clues to the common phylogeny of various species. Humans also have vestigial structures. The small muscles that cause goosebumps were used by our hairier ancestors to signal aggression. Also, human embryos develop tails, which disappear before birth, in most cases. Moving on, anatomical structures that appear similar, that serve the same function, but developed independently in different species, are called analogous.
What's the difference between homologous and analogous structures?
Here's an example. Many birds and insects can fly. They all developed wings in order to fly. Although these functions suggest similarity, they are due to limb bones in the case of birds, but due to parts of the exoskeleton in the case of insects. So they do not demonstrate the homologous evolutionary similarity that a bird's limb bones share with another vertebrate's limb bones.
So different species can adopt in similar ways even when they are not closely related?
That's correct. The kind of evolution that leads to analogous structures is called convergent evolution. Flight may have developed analogously in some birds and insects because of the inherent advantage of flight, but not because they're closely related. An example of convergent evolution is that of sharks and dolphins. Sharks are not closely related to dolphins, since sharks are fishes and dolphins are mammals, but they both developed in such a way as to become successful and agile marine animals. The study of the anatomy of species has led to many insights concerning evolution. However, appearances can be misleading, and what may suggest a common origin could in fact be the result of convergent evolution of very different species.
Thanks for sharing this information with our audience. Today, I would also like to talk about the embryonic development. However, it's fact that related organisms share physical traits.
Do they share anything else?
Yes, they do. The related organism must also share the process that produce those traits. we realize that similar-looking skulls can develop in different ways, depending on how each part grows in proportion to the others. Here is a comparison of skulls. We see that the two human and chimpanzees have very similar skulls, but the two species follow different development pathways. Now let us talk about the comparative embryology.
What do you want to tell us about comparative embryology, Mohammed?
Okay, let me tell you this. The vertebrate embryos appear alike early in development. As development continues, parts grow at different species, and the embryos begin to look less similar. Another thing to talk about today is Ernest Haeckel controversy. The Haeckel admitted that they altered some detail to make the embryos look alike. He also failed to represent the relative sizes of each structure.
I see. Can you tell us what evo-devo is?
Yes, let's talk about it. This term is the study of development genus and determines how mutation can give rise to new body forms. The data shows that there really are similarities in embryonic structures, supporting the concept of common ancestry. Let's have a look now at the early development in fruit flies. Genes are expressed unequally along the length of a fly embryo, setting up distinct environments that profoundly influence development. Given, have you heard about the homeotic genes?
No, I haven't heard about it.
The homeotic gene is a general term describing any gene that relates to organisms with structure in abnormal or unusual places. Also, the homeotic gene is very important because it provides important clues about development.
What does the figure show?
It shows the mutation in these enhancers can cause new features such as wing spots to develop in fruit flies. That's all time we have for this segment. But thanks for being here. We have learned so much.
Thanks for having me. I never missed a chance to talk about biology.
I feel like we are learning so much today on this show. One, I want to touch briefly about how molecules reveal relatedness. To talk to us about this subject is Professor Joan Kamaha, who came all the way from Cameroon, Africa today. Dr. Kamaha, how are you doing?
Well, thank you for having me on the show today. How's everybody doing? Great. Well, as all you know on the show, my name is Professor Kamaha, and I just want to talk about something that we have been taught in school and throughout our life. This is called evolution. Not just evolution in the broad spectrum. I want to talk about how it is important to study our ancestors to be able to understand the similarities and the difference between the modern species and the ancestral species. We as humans are not able to detect and measure the pace of evolutionary change. Crazy, right? But at the same time, it's very awesome. So this is how it goes. We only need about 2 molecules, which are nucleic acids and proteins. We all know nucleic acids are DNA and RNA. With these 2 molecules, one composed of nucleides and the other composed of amino acids, which are the proteins, we are able to trace our ancestry. Did you know that all cells use the same 20 amino acids? Crazy, right? Because of this finding, scientists have found that it is highly unlikely that 2 unrelated species will evolve precisely the same DNA and protein sequence by chance. The similarities between species usually almost come from a common ancestor. These species may look different because through the years some sort of mutation occurred when they broke away from their ancestral group. With molecular evolution, we have realized that you have to look at it from a different perspective. We were taught that you group species and track their ancestry by their similarities. Well, not anymore. Thanks to DNA, we have now been able to open up so many doors of information. Genetic information has helped explain how evolution works. It's crazy, but cells, as you see it, can add new functions to itself by acquiring DNA from other organisms or by duplicating their entire genes.
Proteins, which I mentioned earlier as the second molecule, also help support evolutionary relationships. The study was performed by a group of scientists where they found out that out of 20 proteins, 7 of these were identical in both humans and chimpanzees. Another crazy example is that because of proteins, we have also been able to see that human hair and the keratin, which is a form of protein, in sheep's wool are identical. Now, using keratin is not always effective in performing a study because even though it is a protein, only mammals carry this protein. If you want to study a broader group of organisms, you have to use a form of protein that is present in all species, such as cytochrome. Cytochrome is a part of the electron transport chain in respiration. If you take a moment and look at this picture, you can see that the more closely related two species are, the more alike is their cytochrome. Now, going back to what I had first mentioned in how we can now measure the pace of evolution, did you know that the clock used in this measurement is the one and only DNA? Yes, that's right. DNA itself can act as a clock of measurement. All biologists need to know is the mutation rate and the number of differences in the DNA sequence for that specific gene within 2 or more species. With this knowledge, they can estimate approximately around what time an organism drifted away from its common ancestry and went through mutation change. If you take a look again at this display, you can see a sort of evolutionary clock. Looking at this evolution clock between humans and chimpanzees, we see that about 4 to 6 million years have passed since these two species have parted ways. Another molecule that measures mutation is called mitochondrial DNA. This type of DNA is very important in the more recent evolution between related species because its molecule clock ticks about 5 to 10 times faster than the nuclear DNA. And you can find it more present in extinct specimens such as those displayed in museums. Now, mitochondria DNA is very unique because you can only inherit it through the egg. So that means that you can only get it through your mother. So, in conclusion, if we look at everything that we have learned throughout the years, watching TV, reading books, or even at school about the origin of the first species in Africa, we ask this question: how do they know, and how are they 100% certain? Well, they know this because of mitochondrial DNA, which is the DNA used to trace long-term evolutionary, because unlike nuclear DNA, it does not cross over during meiosis. Using this knowledge, we see that mutation takes time to accumulate, and it shows that Africans have existed longer than any other population.
I feel like I've learned so much today. All of this information is so useful to us as humans because it is always interesting to see how we have evolved throughout times, but also how certain traits and characteristics never change. Thank you so much, Professor, for being on the show today.
No, thank you. The pleasure was all mine.
Moving on to our special guest, Sandra, all the way from Galapagos Islands. She is going to talk about Darwin's finches and evolution. Welcome, Sandra.
Hi, thanks for having me on the show all the way from the Galapagos Islands. It's a beautiful day down here. We got the sun is out, finches are chirping back over here. We have monkeys in the back that are howling. It's just a beautiful day. Hope things in Virginia are warming up for you. As we've learned in previous interviews, fossils' anatomical structures and molecules reveal past evolution. Darwin's finches are a perfect example of evolution. Two ecologists from Princeton, some of my best friends, Peter and Rosemary Grant, conducted one of the most famous studies of these birds. Since the 1970s, they have captured, weighed, measured, banded, and then released nearly every finch on the island. The Grants began collecting DNA from these birds in the 1990s and have measured rainfall, plants, seed diversity, abundance, and size on the island. The goal of their project was to learn more about the 14 species of Darwin's finches and to gather data on individual birds and ecological information to help them analyze the island's short-term natural experiments. They also wanted to document evolutionary changes by comparing multiple generations before and after the event when conditions changed dramatically. One major study was on the island of Delphine Major. they found two permanent birds called the medium ground finch and the cactus finch. The medium ground finch uses its bill to crush and eat seeds of different sizes. The cactus finch uses a probing bill to get the pollen, nectar, and seeds in the petunia cactus. A perfect example of evolution natural selection was in 1992 to 1993 when there was a severe El Nino event that triggered heavy rains for 8 months that changed the variety of seeds on the island. Prior to the El Nino, large, hard seeds dominated the environment, but smaller, softer ones became much more abundant in the following years. This really changed the food source for the medium ground finch because it made it that finches with small bills that could eat smaller seeds were able to eat more than the larger beaked birds. This led to them raising more offspring, which resulted in the average beak width that declined between the mid-1980s. However, the El Nino did not prompt a shift in the size of the cactus finch beaks because their diets did not change and they continued to eat the petunia seeds. The research that the Grants did also really complements a famous earlier study in 1977 that documented a severe drought that resulted in most plants being wiped out. This led to a food shortage and the finches had to eat small seeds that were easy to eat. Only finches that had strong and long enough beaks to crack the seeds survived and reproduced, thus creating finches in the future generations having larger, stronger beaks. The biotechnology revolution has really enabled the Grants and their colleagues to dig even deeper into evolution of Darwin's finches. In 2004, researchers discovered a gene that influenced the development of a finch's cheeks beak. Species with the widest beaks expressed the gene earlier and at a higher level than species with narrower beaks. To confirm the gene's roles in beak development, the researchers increased its expression in a chicken embryo, and the experimental bird responded by growing a wide, deep, finch-like beak. The data that the Grants have collected in the Galapagos Islands clearly reveal that hereditary traits really do change measurably in response to short-term climate changes, and we are further along in the research and theory that Darwin created 150 years ago. I gotta go now. I see a whole bunch of new finches out there.
Sandra, this is absolutely fascinating to see actual examples of evolution. Thank you for your time today. Wow, thank you everyone for coming and teaching us more about fossils and evolution. We've learned so much. I hope that you all have learned as much as I have. Well, thank you everyone for tuning in. Once again, I'm Christine Tyler, and we'll see you next time.